But now let's get started, and I'm very happy to firstly welcome Dr. Claudia Scheckel. She's the CEO of Oncobit, and Oncobit is founded as a medtech spin-off from the University of Zurich. She holds a PhD in biochemistry from the University of Basel and has a degree in management. She worked, among other places, as a biomedical research fellow at Rockefeller University and the University Hospital of Zurich. So thank you very much again, Mrs. Scheckel, for being here today. And I will now hand over to you for your presentation.
Thank you very much for inviting me, but also for this fantastic symposium. It was really great to hear all the talk so far. Can you see 1 second?
Not yet.
Full screen now,
No.
or
no, it's a presentation.
Is that the mode? 1 second. No. Full screen?
Yeah, now it's working. Yes.
Okay, perfect. Great. So thank you again. So I'm Claudia Scheckel. As you heard, our vision at Oncobit is to enable personalized cancer care with our data-driven cancer monitoring platform based on ctDNA with an initial focus on melanoma. And so, as you heard, we are a spin-off from the university and the University Hospital of Zurich. So 2 of my co-founders are actually still professors there. And we actually also started off with RUO, LDT tests, but we thought we want to actually make those tests more widely accessible. And that was really the foundation for Oncobit. And so we are ISO certified, we have regulatory approval for our first product, and we are located just outside of Zurich with our offices and laboratory. And so something that's still very dear to our heart is that we work very closely together with clinicians because we believe that only this way we can develop actually something useful for the patients. And so one example is for example, Reinhard Dummer, which probably several of you know. So I already mentioned we have an initial focus on melanoma. And so when it comes to melanoma, almost really all patients have these hotspot mutations. In cutaneous, it's predominantly BRAF V600 and NRAS Q61. And for uveal melanoma, which is a less frequent subtype of melanoma, it's different hotspot mutations, GNAQ, GNA11, which are mutually exclusive, but also SF3B1 as an additional hotspot. And what's also important is that these hotspots are stable, so they are not lost during disease progression. And that, of course, means that we can follow the patient by just following the hotspot that the patient has. And so one ideal methodology to do so is digital PCR that we've heard a lot about. Because it's essentially just a parallelization of a PCR. So instead of measuring once, you now measure 20,000 times. But this means that you can be more quantitative, more sensitive, and of course you can still detect specific hotspot mutations. And so we have 5 different digital PCR assays developed, and now, of course, they are not LDTs anymore. They actually have to go through very vigorous testing to become a regulatory approved product. With the assays that we have, we cover about 80% of the cutaneous and about 95% of the uveal melanoma cases. The first one is already regulatory approved. The other ones are currently under review. But when we got started with that, we realized that especially when you work with such ultra-sensitive methodologies like digital PCR, a standardized data analysis and interpretation is actually absolutely key. And I think, right, we've heard this a lot earlier today, that when you perform the same test, maybe in different labs, you might get different results. And so this is why our assays are also accompanied by a software, which is also regulatory approved. And so what the software does, on one hand, it automizes everything. So the digital PCR data is read in and it's classified, interpreted, and reported. But importantly, the software was trained on healthy data. And so each of our assay was run on hundreds of healthy samples. And this knowledge is incorporated by the software when it classifies a sample. And so we heard this morning, for example, about clonal hematopoiesis. So we've realized that there are quite some differences on the background, on the noise that you can expect based on the mutation that you're looking at. A second important step that the software does is it performs automated quality control steps. So that means that no matter where the assay is run, the software can correct for a lot of things. And if it can't correct for it, it will flag essentially that the assay was not performed right. And so ultimately, this means that we can report more robust results, but also eliminate, of course, and because of the elimination of a lot of bias. So the way that our system then works is, so we need a minimum of 2 milliliters of plasma. We have a cutoff, and so we also start to quantify at the cutoff, but our confidence intervals depend on the amount of input material. So this is how we this is why we can quantify at every level essentially. But our cutoff, we typically, or also the sensitivity, we always prefer to actually give that not in forms of MAFs, but in the form of mutant molecules. And the rationale for that is that the MAF is, of course, strongly dependent on the input material. And so if you want to standardize this, it's actually much easier to just go with mutant molecules, because then you're independent of the input material. But of course, this corresponds to then, depending on the input, to MAFs as low as 0.01%. And so something that's also that was quite important for us when we started to develop our tests was that hospitals would be enabled to perform the tests themselves. So the biopsy is obviously collected at the hospital, and then the DNA isolation and analysis together with our assays, which consist of a primer probe mix and a positive control, can be either performed in our laboratory, in the laboratory by a partner, or at the hospitals themselves if they have the existing infrastructure. And the reason why we can do that while still maintaining this high standard is because of the software that comes again in to analyze then the data that is uploaded into our cloud infrastructure, which of course fulfills all of the data protection requirements. We don't need a lot of sensitive patient information, right? We just have the digital PCR data. In fact, we don't want any sensitive patient information. It's not needed for us to analyze the samples. And again, this software, because of the built-in QC checks, because of the training that the software has undergone for each of these assays, allows this robust analysis, and then this leads to result delivery. And so with this, we want to provide a very precise monitoring, very sensitive, cancer-specific, quantitative. But at the same time, we want it to be very time and cost effective so that the patient can really be monitored very frequently. And so we heard about a lot of people, like especially like today, started to use these tumor-informed panels, which are very powerful, right? So if the patient doesn't have a hotspot, it becomes very difficult to monitor them with digital PCR. But at the same time, the tumor-informed solutions are quite cost and also time intensive. And let's say if that means that you can realistically only monitor a patient, let's say every 3 months, maybe it's better to use a faster and less expensive method more frequently to kind of compensate for this potentially reduced sensitivity, which in my opinion also still needs to be shown for the tumor-informed approaches. At the same time, of course, there are limitations when it comes to following just single hotspots. And so we also have our own sequencing solution that we usually accompany our studies with. If we want to, for example, look at the coming up of resistant mutations, we typically run this panel. So it has 64 melanoma relevant genes and also resistant genes on it, also HLA types. Again, it's accompanied by a software. And here, of course, yeah, the advantage is if there is no tissue biopsy, we can still diagnose the patient and suggest therapies, identify resistant markers, and depending on which additional mutations are found, also guide studies. We work very closely with the University Hospital of Zurich, specifically with Edle and Reinhardt. And so I just want to share some more recent data on uveal melanoma patients. So they were all stage 4, but so encouragingly, we really found ctDNA in all of these patients that we looked at so far. We also found that with sequencing, we could identify novel pathogenic variants also in actually the majority of the patients, meaning that in the future, this will hopefully also enable then a more personalized therapy adjustment. But at the same time, when we compared sequencing and digital PCR, and in this case, we focused on these 2 mutations, which we can detect with sequencing and with digital PCR. And you see on this plot here, the mutant allele frequency with sequencing and with digital PCR, you see a nice correlation. So our sequencing solutions allows an absolute quantification, but about 1/3 of the samples only had detectable ctDNA with digital PCR. And so We sequence quite deep, but I think in terms of sensitivity, digital PCR still has a lot of advantages compared to sequencing. I want to almost end with one other observation that we, or not rather observation, but I would actually be really interested to also hear the audience opinion on that. So when we report the data, so we heard this morning that that there is not that much guidance on how ctDNA data should be reported, but there's essentially two different ways: the mutant allele frequency and the mutant copies per milliliter plasma. And each of these reporting has its own caveats. So if you can correct for the variance in DNA isolation and assay performance with mutant allele frequency, but if there was, for example, a spike in wild-type DNA, for example, due to physical exercise, right, this leads to an artificially low mutant allele frequency. And so if you report mutant copies per milliliter plasma, that's an advantage here, because there you don't have this correction for this potential wild-type release. And so I would just be maybe in the Q&A very curious to hear your thoughts on this as well. We also looked at cutaneous melanoma, but that's already published. So I think also for reasons of time, I'll skip that. I just want to show the team. So also the team is roughly split into software engineers and assay developers on our side. And we're always looking for collaborations also beyond melanoma to collaborate on clinical studies, also co-development. So if anyone is interested, I'd be happy to get contacted by you. Thank you.